In this next segment, we're actually going to get into kind of the biochemistry. So, we're going to be talking about the types of molecules that make up our organisms. And there are four major types of molecules. We have the carbohydrates, the lipids, the nucleic acids, and the proteins. And all of these are considered organic molecules. So, organic molecules are defined as having a carbon and a hydrogen. So, they used to be defined as just having a carbon. However, if we take a look at carbon dioxide, this is actually considered inorganic molecule, and so the definition then stipulates that you have to have that hydrogen in there. So inorganic molecules may contain carbon but lack that associated hydrogen, as I just mentioned, carbon dioxide. So this is what we're going to be focusing on, these organic molecules, so these carbon and hydrogen containing molecules. We will see that a lot of our organic molecules, they'll have these kind of carbon backbones with some hydrogens, and then they'll be modified with functional groups. So functional groups can contain a OH, which is considered a hydroxide. If a molecule has this, it's usually considered an alcohol. We also have our NH2, that is going to be an amine group. We have a carboxyl group. This is when you have a carbon that is double bound to a oxygen and then has a single bond to a oxygen that is then attached to a hydrogen. That's a carboxyl group. Our ester is going to have a carbon that has a double bond to an oxygen and then has a single bond to another oxygen, and then that oxygen is attached to something else. So whenever you see the R group, it just means something other than hydrogen. So that's considered our ester. Our ether is going to have some kind of group, and then that is single bound to an oxygen, and then another some kind of group that's not a hydrogen attached to that same oxygen. Our methyl group is going to be a carbon that has three hydrogen atoms that are single bound to it. And then our phosphate is when you have a phosphorus atom with a double bond to an oxygen, a single bond to an oxygen, another single bond to an oxygen, a single bond to another oxygen over here, and then attached to some type of R group, some other piece or part, usually this carbon skeleton. Notice that this oxygen here and this oxygen here have a negative charge, and this is because phosphorus is not especially electronegative. So when this oxygen here actually has this double covalent bond, it actually is pulling doubly hard on those electrons. And these oxygens are kind of pulling on those electrons as well. And what we actually see is when this group is actually first formed, it actually has these two oxygens have hydrogens attached to them. But because the electrons from the phosphorus are spending most of their time around this guy here, because he has two covalent bonds, we actually see that the oxygens can't pull as hard on the the electrons associated with this phosphorus, so they actually pull really hard on the electron that's associated with this hydrogen, so hard that they actually steal it, and the hydrogens actually just pop off and form hydroxide ions and kind of float away. So the oxygens, here, let me erase some of this stuff so you can actually see those negative charges again. So the oxygens take on those negative charges. And then we have another functional group, the sulfhydryl group, and this is when you have a sulfur that is attached to a hydrogen. We see this a lot of time in proteins. So when we talk about organic molecules having these functional groups that actually, when we start piecing these things together, they can form biomolecules. So the four biomolecules that we're going to be talking about are going to be our carbohydrates, our nucleic acids, our proteins, and our lipids. So just to give you some ideas of what's going on with these different types of biomolecules, we actually see that carbohydrates, nucleic acids, and proteins can all be what's called polymers. So polymers are formed when we take building blocks or monomers, these little kind of like bricks, and keep stacking them or connecting them in the case of biomolecules together. And when we connect them, they make these long chains. So a building block of carbohydrates can be simple sugars. We can take one sugar, that's a monosaccharide. If we put two of those together, then we have a disaccharide. And then if we just keep adding on to that, then we get what's called a polysaccharide. All three of those are fit under the category of carbohydrates. Nucleic acids, we have in the category of nucleic acids DNA and RNA, and nucleic acids are made up of the building blocks of nucleotides. So more specifically, DNA is made up of deoxyribonucleotides and RNA is made up of ribonucleotides. And we'll talk a little bit more about that when we get to those slides. And then our proteins, those are made up of these building blocks of amino acids. So you piece together all these amino acids, they make this long structure, and that's considered a protein. An example of a protein would be an enzyme. Now, lipids tend to be a little bit different. So they are not technically considered polymers. Instead, there's a couple different categories of lipids that we're going to talk about that are biologically relevant. So there's fats and oils, waxes, and steroids. And we'll talk about those in a few minutes. So let's start with carbohydrates. Carbohydrates are organic molecules that have one or more sugar, as we just talked about. Monosaccharides are actually built from carbon, hydrogen, and oxygen. And the formula for these is actually, oh, my pen's misbehaving again. It doesn't like when I press down too hard. And apparently I get aggressive when I press down. Okay, so let me do it lightly. Okay, so the formula is actually CxH2xOx. Now, what the heck does that mean? That simply means that when you look at a monosaccharide or a simple sugar and you count the number of carbons that are associated with that sugar, you'll actually see that the number of carbons, and if you count up the number of oxygens, those two are going to be equal in number. And then if you count up the number of hydrogens that are associated with that monosaccharide, they're actually double the number of carbons. So a prime example of this would actually be glucose. Glucose is C6H. If we have 6 carbon, it's going to be 12, and then the same number of carbon for oxygen. So C6H12O6, that's our glucose. So whenever you see a chemical formula where you have the same number of carbon and oxygen and double the number of hydrogen, it's pretty much a dead giveaway that that is a monosaccharide. And then these things are going to polymerize, are going to be connected together, and those then, as we connect more and more sugars together, make those polysaccharides. So a disaccharide is going to be two of those monosaccharides pieced together, and they are a disaccharide is formed when those two come together and are chemically bonded, and that particular chemical bond is called a glycosidic bond. So glycosidic bond is associated with our carbohydrates. These are formed via a dehydration reaction, so when these two come together, we actually see water is also formed. So let's just say you have two glucose monomers and you want to form a disaccharide with these two glucose monomers. Well, you're going to see that when you form those, the chemical structure associated with the formation of these two glucose monomers coming together is actually not going to be C12H24O12 because in the formation of that glycosidic bond, you lose a water molecule. So if we want to determine the actual chemical structure of the resulting disaccharide from bringing two glucose monomers together, it actually would be C12 and then 24 minus 2 for that water molecule would be 22, and then oxygen we have 12 but minus the 1 for water give us 11. So that would be the actual chemical formula here for sucrose. So we actually combine those two together. So what's the function of carbohydrates? I don't want you guys to have to know specifics, but I do want you to know this kind of list here. So carbohydrates can be used as energy sources. They also can be used as structural biochemicals. They're used in cellular adhesion, in extracellular matrices, allow the cells to kind of stick together. They also play a role in communication and then also play a role in environmental sensing. So they have quite a diverse array of functions. And as we start talking about the actual prokaryotic cell structure and eukaryotic cell structure, you'll actually, and also metabolism for energy sources here, you'll actually better understand how carbohydrates function. So some examples of carbohydrates that I want you to be familiar with. This is obviously by no means an exhaustive list, but these are some of the major players when we start talking about biological molecules for our cells. Cellulose is the most abundant polysaccharide on the planet, and this is the major structural macromolecule in plant and algal cell walls. So it's actually a polymer that helps create the structure associated with plant cell walls and in some cases algal cell walls. Chitin is a structural polysaccharide that is specifically found in fungal cell walls. And then peptidoglycan, if you remember chapter 1, I told you when you think of bacteria, think of peptidoglycan. So peptidoglycan helps form part of the bacterial cell walls. And just remind yourself, gram positives have that really thick layer of peptidoglycan, whereas our gram negatives have that thinner layer. We also see that capsules are a carbohydrate-based structure, and these are made by some bacteria, not all. It does a couple different things. It actually allows them to adhere to surfaces, but it actually also it coats the bacteria. So this is an example of Streptococcus pneumoniae. Streptococcus pneumoniae is actually Diplococci bacteria. So there's two little bacterial cells that stick together, and then they both form this carbohydrate substance that surrounds them, and it actually protects them from our immune system. So our immune system basically cannot recognize them. So it also gives them protection. Okay, on to our lipids. Our lipids, as you recall from the beginning of this conversation, I said these are not going to be polymer based. So there's actually a couple different types of lipids that I want you to be familiar with. So lipids include our fats, oils, waxes, and steroids. They are predominantly hydrophobic. So remind me, what does that mean? That means they hate water, so they're not going to really interact with water. But we do see that a couple examples of lipids are amphipathic, meaning that they have parts that are hydrophilic and parts that are hydrophobic. And we'll talk about which ones those are as we move forward. And we do see that in this category of biomolecules, there's a lot of diverse structures, and they can range in size and complexity. So we'll talk about that as we move on. So when we look at lipids, we see fats and oils are usually built by combining what's called a glycerol molecule, and that is actually pictured here. So glycerol actually has 3 carbons that are associated with it, and attached to those carbons are some hydrogens, and then you'll also see this oxygen here, here, and here, and then that usually has a hydrogen attached to it, but when we actually form these covalent bonds with these other parts of the actual fat, we see that we'll lose that hydrogen along with an OH group from this other component, forming water. So these interactions, these bonds here, are actually formed via a dehydration reaction. So when we see our fats being formed, this glycerol molecule is attached to fatty acids. And there's 3 attachment points. And each of these attachment points is an ester bond. Now our fatty acids that are attached to our fats, attached to the glycerol to make our fats, we actually see that we can have one or more attachment sites to that glycerol molecule. So if there's only one, that's considered a monoglyceride molecule, and it only has one of those fatty acid chains. We can have 2 attached. That's a diglyceride, and it has 2 fatty acid chains. And then our triglycerides have 3 fatty acid chains. Triglycerides are really, when we say the word fat, this is actually what we're talking about. We're talking about triglycerides. Now, when we look at those fatty acid tails, we actually see that they can be categorized based on what's going on with these carbons and the associated hydrogens. So if our carbons have a bunch of hydrogens and we don't see any double bonds associated with the carbons in this fatty acid tail, this is considered a saturated fatty acid. And that just simply means that that carbon is saturated with all hydrogen atoms. So they're going to lack any double bonds and they're going to be pretty straight. What's interesting is that these are fats that tend to be associated with animals. So animal fats, so butter and lard is an example of these types of triglycerides with the saturated fatty acid tails, and they will tend to be solid at room temperature. So a kind of easy way to think about this, if you've ever went to say like the meat counter and you're looking for a steak, a lot of people that are kind of connoisseurs of meat, they will look for marbling. That marbling is white. Mixed in with the red of the meat fibers. The white is actually fat, and that's what gives the moisture and some what some people think the taste and the flavor to the meat. So that white is solid, so that is actually an example of a triglyceride. Now we can also have unsaturated fatty acid tails, and when these unsaturated fatty acid tails come together, we actually see that the carbons are not saturated with hydrogens, and we'll find a double bond in that tail. And that double bond will usually introduce a little kink in the tail, and that actually causes them to be a little bit more flexible, and they won't actually pack in really close together. So they tend to be a lot more liquid and viscous. So some examples of this are going to be like oils, olive oil, corn oil. So these are a lot of times associated with our plants. So they'll have those kinks and bends that allow them to then be a quote unquote liquid at room temperatures. We also have waxes. Waxes are going to be molecules that contain fatty acids that are linked to a long chain alcohol. And remember, an alcohol has that hydroxyl group associated with it. So here's just an example. This is beeswax. So here is your fatty acid, and then here's your long chained alcohol. Notice we don't talk too terribly much about how we draw these, but these little lines here are indicating that you have carbons and they're saturated with hydrogens. So notice that this molecule is predominantly carbons and hydrogens, which are predominantly nonpolar covalent bonds. So this is going to be a hydrophobic molecule. And in fact, we do see that a lot of organisms actually will make waxes so that they become resistant to water, or they can repel water. Example of this is ducks, birds that like to be on the water. They will produce a wax on their feathers so that it helps them stay buoyant and helps them stay dry as they're hanging out in the water. Steroids are another type of lipid, and what's interesting about steroids is they actually contain four fused rings. So here is one of those rings. There's the second, there's the third, and then there's the fourth. And when we take a look at these rings, we actually see that three of the rings actually are six-membered rings, so little hexagons. And then this fourth one is actually a five-membered ring, so a little pentagon. Now, we actually can add different functional groups to different places on these fused rings to give us different types of molecules. So just to give you an example, there's our base, our four fused rings that I've already kind of scribbled over. And if we add this hydroxyl group, a methyl group, and then a oxygen, double bound to the carbon there, we actually form testosterone. Now we can take this and add an OH group here, a methyl group methyl, and then add this branched molecule or branch-like functional group, and that then forms cholesterol. So sterols are modified steroids with attachment of alcohol functional groups, and cholesterol is going to be an example of that. So all of them kind of have that basic foundation of the four fused rings. We start adding new things in there, it gives them new properties and allows them to do different things. So what are the functions of lipids? Well, they actually are great for energy, and again, we'll talk more about that when we get into metabolism. We also see that they can play a role in cell structure components, and they can help mediate cell signaling, and we'll talk more about those as we move forward. Some prime examples of our lipids, phospholipids. So this is a really big one. So phospholipids have the hydrophilic head and those hydrophobic tails. So they come together to form those bilayers, and you get enough of them, they form an actual sphere, and that then forms the cell membrane, also known as the plasma membrane. So they are amphipathic and they do form those bilayers. We also have glycolipids. Glycolipids are lipids and oils that link to carbohydrates. We'll find these are a lot of times associated with the plasma membrane. And lipoproteins, these are lipids and oils that are linked to proteins. And again, we'll find these actually associated with the plasma membrane. So here's a little plasma membrane that is actually made up of the phospholipid, and the phospholipid is attached to a protein for the lipoproteins, or in the case of our glycolipids, it'll be a carbohydrate that's attached to. And we'll again talk more about this when we get into our conversations about eukaryotic and prokaryotic cell structure. Another really important one is going to be our lipopolysaccharide, or LPS. Now remember when we talked about the structure of gram-positive versus gram-negative cell walls, I told you all that the gram-positive has a thick layer of peptidoglycan. Okay, great. We already talked about that one. That's a type of carbohydrate. But then our gram-negatives, they actually have that thin layer of peptidoglycan, and then they have that outer membrane. And that outer membrane, one half of that outer membrane is actually got lipid associated with it, and then it's got a core polysaccharide. And this is actually the LPS component associated with the gram-negative outer cell wall. And again, we'll talk a lot more about that when we get into our prokaryotic cell structure. Mycolic acid, that is the part of the acid-fast bacterial cell wall. So remember, our acid-fast cells, they actually, if we were to strip that mycolic acid, they would present as gram-positives, but then they have that thick layer, waxy layer of mycolic acid. Oops. And this actually allows them to increase their pathogenicity. So that's just a fancy way of saying that they are better at causing disease, because this waxy mycolic acid cell wall protects them from the immune system. And then cholesterol. I know a lot of you, when you think of cholesterol, you're like, "Ooh, cholesterol is bad for you." Well, actually, it's really abundant in animal, and we're humans are considered animal, cell plasma membranes. So our actual plasma membranes, if we take a look at those phospholipids and how they form the bilayer, we actually see tucked in there will be those little cholesterol molecules with the four fused rings, and then the branched functional group attached to it. So they kind of act as like almost like kind of packing peanuts, if you will, in animal cell plasma membranes. All right, moving right along to our nucleic acids. Nucleic acids are our macromolecules that serve as genetic material of cells and viruses. So specifically in nucleic acids, we have DNA and RNA. DNA is short for deoxyribonucleic acid, and then RNA is short for ribonucleic acid. In general, DNA tends to be double-stranded, and it tends to be helical. So when we look at a DNA molecule, it's usually drawn like so. Again, if my pen would cooperate with me. Whereas our RNA tends to be a single-stranded molecule, but what's interesting about the RNA is a lot of times RNA self-anneals, so it'll kind of fold back on itself in some cases. Our nucleic acids are polymers of nucleotides, as I mentioned before. And those nucleotides, if we take a look, and this is just the generalized nucleotide, we see our nucleotides are made up of a sugar, and they'll have a phosphate group attached to that sugar, and then they also have a base, and that base is where each nucleotide will differ. Now, when we're talking about DNA versus RNA, we actually see the difference in the nucleotides comes from the sugars themselves. So in RNA, we find the ribose sugar. And if we take a look at the sugar, we actually see that it is circular here. So it has 5 carbons. So there's 1, 2, 3, 4, and 5s right up there. And if we take a look at the sugar that makes up DNA, it's called deoxyribose. Now, deoxy, the prefix D, basically means lack of. Let me erase that so you can actually see this. If you take a look at carbon 1, 2, let's number them 2, there's 3, there's 4, there's 5, so there's 5 carbons. But look at carbon number 2 here versus carbon number 2 in the ribose sugar. See how it's missing that oxygen? So deoxy basically means removal of oxygen. So that's the major difference here. The deoxy removal of oxygen on that second carbon. So that is actually missing from the sugar that makes up DNA, whereas we find that oxygen in that ribose sugar that makes up RNA. We see that there tend to be, when we look at nucleotides in the actual DNA polymer or RNA polymer, it only has one phosphate group, but the precursors to this actually contain up to 3 phosphate groups. And we'll talk more about that when we get in our genetics chapter. We'll talk about the process by which DNA and RNA are synthesized. Okay. This base is going to dictate what type of nucleotide we're looking at. So the base is going to differ. And the base is nitrogen containing, so we call them nitrogenous bases. And we'll have cytosine, uracil is found in RNA but not DNA, thymine is found in DNA but not RNA, and then guanine and adenine. So cytosine, guanine, adenine, all found in both DNA and RNA, but uracil is specific to RNA and thymine is specific to DNA. So these are kind of more the special ones. So if you're looking at a sequence and it says, okay, we have A, T, C, and G, then you know that's going to be DNA because you have that thymine. And if somebody says, okay, I have this nucleotide, or excuse me, this nucleic acid, and it's A, U, G, C, then you know that you're working with RNA. All right, so our DNA, as I mentioned, made up of our deoxyribonucleotides. It has that deoxyribose sugar and has the nitrogenous bases adenine, guanine, cytosine, and thymine. Whereas our RNA made up of the ribonucleotides, and those ribonucleotides contain the ribose sugar, and the nitrogenous bases associated with that are going to be adenine, guanine, cytosine, and uracil. So this is kind of a summary slide for us. Both DNA and RNA nucleotides are going to be connected to each other in the same fashion, not very similarly, actually in the same fashion. So we actually see that you have your nucleotide number 1 here, and it's going to come in and connect to nucleotide number 2. So when we take a look at these nucleotides, let's go back to counting these carbons. So this is carbon 1, 2, 3, 4, and 5. Attached to that 5th carbon is the phosphate group. So this is actually referred to as the 5' end of this particular nucleotide. And always at the 5' end, we're going to find a phosphate. And then when we look at the carbons again, we actually see that this chemical bond formation occurs at this 3rd carbon. So this 3rd carbon is referred to as the 3' end, and at the 3' end we're always going to find a hydroxyl group. So whenever anybody talks about 5' end of DNA or RNA, they're talking about the end that has the phosphate, and when they're talking about the 3' end of DNA or RNA, they're talking about the end that has the hydroxyl group or the OH. So when these form this connection, we actually see that this carbon comes in here and attaches to the oxygen associated with the phosphate group, and we have the formation of this bond. Notice we actually are going to lose the hydrogen and the OH here, so this is actually considered a dehydration synthesis. So we are going to see that we're going to lose water in this dehydration reaction, and that leads to the formation of a phosphodiester bond. As we have these being added in, notice what's happening here. All these bases are kind of sticking out here, and then we have this kind of repeating almost backbone associated with this molecule. So you have phosphate, sugar, backbone. And so this is actually referred to as the backbone of the molecule, and it actually is referred to as the sugar phosphate backbone of the molecule. So what's the importance of our nucleic acids and what do they do? How do they function? Well, they're kind of, if we're talking about DNA, it's the instruction manual of the cell, so genetic blueprint of all of our cells. We also see that DNA in most of our cells would be nothing without RNA, so DNA kind of acts as an instruction manual to make RNA, and RNA then acts as an instruction manual to make proteins. We also see that there are special RNAs that can catalyze certain reactions, and those are referred to as ribozymes, and we'll talk a little bit more about those when we get into metabolism. And then the precursors associated with nucleotides. So I showed you guys where you have your phosphate and your sugar, and then the base. Well, the precursors to nucleotides actually have additional phosphate groups. The fancy name for these are nucleoside triphosphates. And again, don't worry about that right now. I just wanted to point this out to you for the purposes of bullet number 2 here. So most ribonucleotides can actually serve as energy sources. So you might be familiar with the term ATP. Well, ATP is simply the precursor for adenines in RNA. However, it also can be used as energy. So you can actually strip off these terminal 2 phosphate groups and that releases energy. So ATP can be broken down into ADP. Oh, excuse me, you strip off these 2 phosphate groups if you want to make the nucleotide for DNA and RNA. But for the release of energy, you can strip off one of those phosphate groups to make a ADP and inorganic phosphate. And then you can also, depending on the enzyme, you can actually strip off the other phosphate and make AMP for monophosphate, and then you remove that other terminal phosphate group and you get energy out of this. So all of that gives you energy. All right, our final biological molecule that we're going to talk about is proteins. So as I mentioned in the previous slide, DNA is the instruction manual that gives rise to then RNA, and RNA then gives rise to proteins. So DNA basically indirectly mediates the production of proteins. And at some level, every cellular process involves a protein. And in fact, proteins are so important in the function of cells that if you stop protein production in a cell, it will die. So no proteins, no survival of that cell. And we actually see that there are some antibiotics who target the synthesis of proteins in bacteria in order to kill off that bacteria, because we know that no protein synthesis, no cell survival. A lot of proteins you'll actually see illustrated kind of like this figure here. So you'll see, and we'll talk about the structure of proteins and what this all means, but these little helices, and then if you notice these little strands here that have arrows associated with them, those are referred to as beta pleated sheets, and I'll explain a little bit more about protein structure as we move forward. So proteins are polymers of amino acids. And when we look at the number of amino acids, we actually see that there are 22 that are genetically encoded for. 20 of those are your standard amino acids, and then there's 20 non-standard amino acids. So they have to go through some modifications. Each of our amino acids consists of a carbon that is attached to other functional groups. So this carbon will be attached to a hydrogen and then also has an amine group, which is going to be the nitrogen with 2 hydrogens attached to it, and then also a carboxyl group. So every single amino acid has this general structure. So how do those 22 amino acids differ? Well, it's through what's called the R group. So that's where we have variation from amino acid to amino acid. So if you're curious, here are some examples of R groups that are possible. Now, I don't want you guys to memorize all these different structures. That's, if you continue on in biology, you will take a biochemistry class and they'll make you do that in biochemistry, but it's more of a 4,000 level course requirement than this. I just want you guys to know that we have non-polar amino acids and we have polar amino acids, and then that can give rise to different types of functionality associated with amino acids. So this shows you that those R groups can actually be as simple as, say, a hydrogen or a methyl group to a more complex fused ring structure. And these are all examples of nonpolar amino acids. And then we have our polar amino acids. So these are going to have polar groups associated with them. And again, you guys don't have to memorize these. I just want you to be aware that there are nonpolar and there are polar amino acids. There are also acidic amino acids. So acidic amino acids are negatively charged amino acids. So they will usually lose this hydrogen and take on a net negative charge. And then we also have basic amino acids. Those will take on positive charges associated with that amino acid. And then here are those non-standard amino acids. So amino acids are connected to each other by peptide bonds. With peptide bonds, an amino group of one amino acid is attached to the carboxyl group of the next amino acid. So this means that we're going to have a functionality, or excuse me, a directionality associated with our proteins as well. So one end is always going to be an amino end and one end is always going to be our carboxyl end. Peptides are the name given to short little amino acid chains. Polypeptides are really long polymers of amino acids. And these polypeptides then undergo folding, and they are not really considered proteins until they are properly folded and have a biological function. So proteins are kind of like the term that's given to these polypeptides once they are ready to go and ready to function. Now, when we have protein folding, we actually see that it's relatively complex and there's multiple levels to protein folding. So we're going to talk about the different levels and the different levels of protein structure. So we have primary, secondary, tertiary, and quaternary. So primary structure is simply the linear sequence of amino acids. So let's say I had a tryptophan here, and then I had a glycine, and maybe cytosine, maybe a proline, so on and so forth, just listing out the amino acids that are in that particular protein. That is your primary structure. Secondary structure is when you actually have interactions between the amino and carboxyl group. So here's that carbon, you have your amino and carboxyl group, and then you have your little hydrogen, and then you have your R group. So we actually see that in some instances, the strands of the polypeptide will kind of curl around each other, and your carboxyl and your amino groups associated with this backbone of the amino acid will actually interact. And that is actually what then leads to the formation of the alpha helix. So you can actually kind of see that it's spiraling around. And then in some cases, we'll actually see that these polypeptides will kind of curl around and we'll have interactions between the amino groups and the carboxyl groups of amino acids that are on adjacent strands. And that then leads to the formation of these beta pleated sheets. So again, I just want to point out this is going to be associated with the backbone of those amino acids, not the R group. When we have interactions among the R groups, that's what actually establishes the tertiary structure. And a lot of this leads to three-dimensional folding in structure. We'll have non-covalent interactions that occur, so ionic bonds. So remember I told you guys that some of those amino acids could be acidic or basic, so they have negative and positive charges. Well, if you have a positively charged amino acid that comes into close proximity with a negatively charged amino acid, guess what? Ionic bond forms. We can also have hydrogen bonds forming with the polar amino acids, so the partial positive and negative charges can come together, and then our non-polar amino acids can interact via van der Waals interactions. And we do have, in some cases, our cytosines come together and they actually have sulfhydryl groups. And when those come into close proximity, we'll actually see that the amino acids will lose the two hydrogens. Let's see if I can erase those two hydrogens, and then we'll have a covalent bond that actually forms between the two sulfurs. So this is two separate amino acids that then form this covalent bond. That's referred to as a disulfide bridge. So all of those different interactions then lead to tertiary structure, which causes protein basically to kind of fold up. And then we can have quaternary structure. This doesn't necessarily arise with every single protein, but if we have one protein that is made up of more than one separate polypeptide, when those separate polypeptide chains come together and interact, so you can kind of see this is kind of a dark purple color here, and this is a light color purple here, and you can see how they're kind of interacting at different points. That is our quaternary structure that is then formed. So what are the functions of proteins? Well, cellular life, there's a bunch of different things they do. So they can act as structural components of the cells. They can lead to signaling within the cell. They can act as enzymes and then facilitate those chemical reactions. They can act as transporters within the cell. They can be on the outside of the cell and act as kind of like little molecular tags that allow for cellular recognition. They can be released from cells and bind to other cells and aid in cellular communication. So they have a mirage of different, a myriad of different proteins associated with them. So that then ends our conversation on biomolecules. And again, we'll revisit exactly what these things are doing as far as structure and function when we start into week 3's topics on prokaryotic and eukaryotic cell structure. So if you have any questions, please don't hesitate to ask. And I definitely encourage you to watch the next video, which will deal with some challenge questions for you or some think about it questions for you.